My name is Moepeng Maseko. I'm one of the virology registrars. So my presentation today is about, the title is No Amateur Virus. So it was inspired by this case. I had a case of a 35-year-old female who is known with HIV on first-line antiretroviral therapy of tenofovir, efavirenz, and emtricitabine since 2009. She has now been documented as virological failure, although she is insistent that she has been adherent to treatment. She's known as iron deficiency anemia. She's got a history of recurrent TB meningitis and is currently now on treatment for the second time. She presented to the hospital feeling unwell with worsening fatigue and night sweats. She has no family history of any other illnesses. And upon seeing her, they repeated her CD4 cell count and her viral load, which showed a CD4 cell count of 4,178 and a viral load of 2,850. So the doctors, upon seeing this, did more bloods, but the ones I could access were the white cell count, which showed a progression of increasing trend of white cell count and the iron deficiency anemia there, but it was markedly increased. So because of the very, very high CD4 cell counts, they discussed to the patient with hematology who recommended doing a HTLV1 screening. So they did an HTLV1 IgG, which is done in KZN, and it came back positive. So they sent us a sample so we could do a confirmatory test for HTLV1, which we do a PCR, which came back positive. So they were suspecting malignancy in her, which is why they discussed with heme. So my case today is about human T-cell lymphotropic viruses. So these viruses belong to the Retroviridae family of the genus Delta retrovirus. They are linear, single-stranded, positive sense RNA viruses, and they are the only known human oncogenic retroviruses. So there's 4 different HTLV viruses that are known, but only HTLV1 and HTLV2 have been linked to human disease. In terms of the geographic distribution of the subtypes as well, HTLV occurs worldwide, but is more prevalent in certain areas such as Japan, South America, Australia, Sub-Saharan Africa, Middle East, and the Caribbean. And it's estimated about 10 to 20 million people are infected worldwide. So as we can see, the different subtypes conglomerate here around Central Africa, which is where the primates are, and which is how we got HTLV in the first place. And then the rest of the world is dominated, or the areas that are affected are dominated by the transcontinental subtype because of traveling between continents. And then this prevalence closer to home. In terms of the prevalence, studies have been done have been mostly based on blood donors, pregnant women, and hospitalized patients. But a summary of 2019 summary of published reports in Sub-Saharan Africa, blood donors showed rates of occurrence that range from 0 to 5%, with an overall rate of 0.68% for HTLV1 and 1.1% for HTLV2. So that was that for this. But in 2018, the South African National Blood Service did a seroprevalence study in their blood donors to determine the risks posed by blood transfusion. And this was in part triggered by a 2014 case report of a 65-year-old man who presented with progressive lower limb weakness and was later found to have transfusion-acquired HTLV. So this study by the South African National Blood Service found a 0.12% positivity rate with a statistically significant difference in positivity rates amongst age group, gender, and race. And the highest prevalence was in Black donors at 0.16. So through extrapolation to the total blood supply for the South African Blood Service, it estimated that prevalence rates to be at about 0.062%, which translates to about 509 potentially infectious units of blood annually. And the estimated prevalence in the adult Black population was found to be about 1%. So in terms of the geographical distribution of the prevalence study from that study, it showed no significant difference in the prevalence amongst the operational zones of the South African National Blood Services, except that there was no HTLV found in the EC, the Eastern Cape. Well, they explained in the study, they explained that they had practiced over sampling of certain races, which was the black race and the mixed race, because they predicted that they would find a higher prevalence rate in these races, which they seem to have. But now they can't extrapolate to the rest of the population because it doesn't include any white people in it. And then in terms of figure 2, it's now focused on the black population, showing that prevalence increased with age, especially above the age of 50, and females were more likely to be positive than males at all ages. Okay, so the modes of transmission of HTLV, besides the already mentioned parenteral route from blood transfusion, you can have mother-to-child transfusion or sexual transfusion, mother-to-child transmission or sexual transmission, where women are more susceptible. And there's also infection with HTLV1 among drug users, but to a lesser extent, the HTLV2 is more than HTLV1. I hope that makes sense. All right, then we get to some interesting characteristics of HTLV1. So HTLV1 preferentially infects CD4 cells, but because of the universal nature of its cellular receptors, it can infect other cells, including CD8 T cells, dendritic cells, and macrophages. It's also a retrovirus, but unlike HIV, HTLV1 infection expands by immortalization of infected cells that proliferate. And instead of killing CD4 cells to induce immunodeficiency, it alters the function of immune cells over decades to cause associated diseases. Okay, then we get to the pathogenesis of HTLV1. So in terms of its pathogenesis, so it starts off, well, its main mode of replication is clonal. Well, it starts off by clonal integration into CD4 and CD25 T lymphocytes. And after transmission, it increases its viral copy number by both lytic replication and mostly clonal proliferation of infected cells. So the proviral load reflects immune control, and complications are usually associated with a high proviral load. So TAX, which is one of the regulatory proteins of HTLV1, is a transactivator. So it activates viral transcription and therefore replication, but it is also very immunogenic. So the more TAX is being expressed by immune cells, the more the immune response will clear these cells. So to counteract this, HTLV1 codes for another regulatory protein, which is called HBV. And this then downregulates the expression of TAX, and this assists in shielding the virus from the immune system, but upregulates T cell proliferation and therefore HTLV replication because of the genome of HTLV1 being embedded in the T cells. Now, this figure here shows the natural cause of HTLV1 infection to the onset of adult T cell leukemia. So it starts off here with a HTLV1 infected cell will infect another cell by cell-to-cell transmission, and then it will result in clonal proliferation. Here it looks like a monoclonal, but it's usually an oligoclonal proliferation, where after a long latent period, ATL will develop in approximately 5% of asymptomatic carriers. This is believed to be partially due to alterations and errors in the host genome, which accumulate progressively during the latent period, finally leading to the onset of ATL, which is adult T cell leukemia. Right. Then we have the clinical symptoms that are associated with HTLV1. I've mentioned one, which is the leukemia. The two most common are the leukemia and the myelopathy. So in terms of the leukemia, you have a CD4 T cell leukemia, which occurs in about 3 to 5% of all carriers, and in about 20% after infection around the first time. It usually develops after the third to the fifth decade of latency. And then the other common one is a HTLV1 associated myelopathy or tropical spastic paraparesis, which occurs in about 0.3 to 5% of all carriers. And that's usually after 2 to 4 decades of latency. Other clinical symptoms that are associated with this would be a T-cell immunodeficiency, where it can also be seen in asymptomatic carriers, but patients who present with conditions such as bronchiectasis, and children can present with superadded infections such as impetigo because they just lack cell-mediated immunity due to this infection. And then the inflammatory conditions that can result from this would include uveitis, all the arthropathies such as rheumatoid arthritis, the infectious dermatitis, myositis, all those inflammatory driven pathologies. All right. Okay, so in terms of HTLV1 and HIV co-infection. So around the world, co-infection is mainly reported in South America, the Caribbean, and Africa. These two retroviruses are opposite in many aspects. So HIV replication is vigorous in vivo, which is in contrast to HTLV1, which increases its copy number mainly through proliferation of HTLV1 infected cells. So thus although HTLV1 polymerase is error-prone, most of the replication of the genome is via the high-fidelity post-DNA polymerase. So therefore, there's much less variation in the HTLV1 provirus sequence compared with HIV. But however, because T-cell immunity is critical for controlling the number of HTLV1 infected cells, HIV-infected patients are at increased risk of developing HTLV1-related pathology. So some publications describing co-infection complications in South Africa noted the following. The first one, which is the top study there, was a study that was done in KZN in 2017, which compared HAM, or HTLV1 associated myelopathies, or TSP, in HIV positive and HIV negative patients. So the general findings were that co-infection may accelerate the development of TSP in patients, which results in them presenting at a younger age. And the median age was 31 years versus 50. And then in that study, they found that CD4 T cell counts were preserved with a median of 781. The bottom study here did the same comparison. It was done in Steve Biko Hospital, where co-infected patients were also found to present sooner, but they also found an extra finding of thoracic cord atrophy, which was very common in both groups. 7 out of 8 co-infected patients versus 8 to 10 mono-infected HTLV1 patients were found in that study. All right. So HTLV1 co-infection, okay, so as seen with the case, there was not much detail in that case, but it seems like the patient was heading towards malignancy of ATL, but it was just a struggle finding the rest of the blood results. But the effect of co-infection seems to converge with a worsening of symptoms linked to HIV and HTLV1 infection by one, in terms of HIV, accelerating the progression to AIDS, or increasing mortality, because co-infected patients usually have a significantly higher cell count, which are identified as CD4 plus T cells, but they're non-functional. So this drives HIV replication and complicates the assessment of the level of immune suppression. And then co-infected patients are more likely to suffer from myelopathy, thrombocytopenia, and infectious opportunistic infections, regardless of the age or CD4 T cell count. In terms of the HTLV, there's been reports that have suggested that co-infection can influence the development of HTLV1 associated diseases, with HIV worsening the clinical outcome of HTLV1 infection. All right. Then we get to how do we diagnose this HTLV1? So the seroconversion period has been reported to be as long as 65 days, but there's been cases of delayed seroconversion which have been reported. But there's infants that are born to seropositive mothers have been reported to seroconvert within 1 to 3 years of age. So in terms of testing, we can do an antibody, which is serology-based testing, or molecular methods for testing. In terms of antibody detection, ELISA is the most frequently used screening test, which tests for HTLV1 and 2 antibodies. It does not discriminate between the two. And then a Western blot is normally used for confirmatory testing, which also distinguishes between infection with HTLV1 and 2, which is the less pathogenic. And then in terms of the molecular testing, proviral DNA detection can be done on both whole blood and cerebrospinal fluid, and if done here in the NHLS on an antibody positive predictive value. So the PCR-based testing that we do detects proviral DNA in peripheral blood mononuclear cells, but it only detects HTLV1. We do not detect HTLV2. All right, so diagnosis in the proviral load. So quantification of the HTLV1 provirus among infected individuals demonstrates that a high proviral load is associated with symptomatic versus asymptomatic individuals. So asymptomatic patients usually have a typically low proviral load set point relative to those that have conditions such as TSP. And the higher number of infected cells is also thought to facilitate transmission, particularly in the proviral load in breast milk. And longitudinal studies show that HTLV1 proviral load remains steady over time in patients with CAM and TSP and in asymptomatic carriers. So in terms of treatment, the viral replication inhibiting antivirals are not available. So there have been limited studies of specific HIV antiretrovirals that inhibit the HTLV virus transcriptase that have been assessed, but they are generally not indicated because HTLV1 pathology is mainly driven by clonal expansion of infected cells. So, but the specific ones that have been done, AZT and interferon alpha have been used in clinical trials with some benefit in patients with ATL. This was unknown whether it was due to the antiviral activity of the AZT and, or due to direct toxicity. So these two have been shown to increase the 5-year overall survival rate when used as first-line antiviral therapy compared to chemotherapy, but decreased the survival rate when used after chemotherapy. But this is only seen in some types of ATL, which is the acute, the chronic, and the smoldering, not the lymphoma-based subtype. And the optimal duration of treatment is not yet known, so, and this is only been tested in clinical trials. In terms of other antiretrovirals, so lamivudine and cidovudine has been used in patients with HAM. There have been no significant differences in clinical parameters or proviral load between the treatment and the placebo groups. And when lamivudine was used alone in the same patients, there was a tenfold decline in the DNA initially, and fluctuations between baseline and the lowest point thereafter. So it seems to be very successful, and they could not really explain why this was the case. So what is the treatment that patients do get? So in terms of the tropical spastic paraparesis, it's mainly symptomatic treatment to treat the signs of inflammation. So we treat with corticosteroids and prednisone. And in terms of adult T-cell leukemia, chemotherapy is the main mode of treatment. First generation chemotherapy was used, which is the CHOP. It was used, but it had poor outcomes. So then the lymphoma group study 15 LSG-based regimes have proved to be more efficient as a regimen, which is and then there's also been some anti-CCR monoclonal antibody has been registered, which is licensed in Japan, has also been tried. So the CCR4 chemokine receptor is overexpressed on HTLV1 infected cells in approximately 90% of ATL cases. So a positive CCR4 in an ATL cell has been associated with an unfavorable prognosis. So this anti-CCR monoclonal antibody induces a strong antibody-dependent cellular toxicity because of its high affinity binding to the effector cells. And then monotherapy showed clinically meaningful anti-tumor activity in patients with aggressive ATL who relapsed after they've gotten chemo. This results in a combination of therapy with chemo for aggressive ATL. But if you are to use considering allogeneic hematopoietic stem cell replacement, this anti-CCR monoclonal antibody cannot be used. So basically, well, I mean, overall, there is poor prognosis for the treatment of these conditions. And that's my story.
Thanks very much indeed for a very comprehensive presentation, beautifully made. would you like to just stop sharing your screen? And maybe I could ask our virology colleagues, consultants, to perhaps to give their views. So Di or Marvin, would you like to give some input?
So maybe just to start off with, so
could you pop your video on, Di? Sorry.
Oh, I'm afraid I don't have a video on this particular
Okay, don't worry.
PC. Yeah, so we run this. It's. I think this is a diagnosis, or certainly in the Western Cape, it's not a condition that is very frequently considered, and probably there's more pathology out there than is necessarily recognized. We've been running doing the HTLV1 PCR for now quite a few years, and it's really, we really offer it as a kind of as a confirmatory test. I mean, serology is available, but only because of the sort of relatively low low prevalence of infection. The sort of it's useful to have a confirmatory test, and yeah, and so we tend to get samples referred from around the country. I would say mostly they are patients either with neurological presentations, and then the occasional probable T-cell leukemia. Yeah, so I think it's a virus that probably it's worthwhile to bring it back into the sort of the you know, the differential of these yeah, conditions.
Okay, no, thanks very much. Marvin, while you're considering what you'd like to give input in, I mean, so my understanding, well, and firstly, we tend to think of this virus with HIV in the setting that you've detailed in your presentation, which relates to patients who are presenting with very high CD4 counts when they shouldn't really have them, and particularly CD4 counts over 1,000. Previously, the teaching was that tropical spastic paraparesis, or the HAM, was much more common in the Caribbean, and the leukemia forms were much more common in Africa. I'm sorry, you may have told us that, but I may have missed it, but is that still the case? Or is that not the case? Could somebody answer that?
I didn't actually mention it, but I mean, I think it's interesting that you mentioned that. We've had, I think over the past month, we've had 2 patients that have been referred to us who were query T-cell leukemia, but I'm not sure of that. Maybe one of my consultants.
Yeah, but there's going to be a bias there, obviously, because you've got a bone marrow unit here. Di or Marvin, are you able to help us with that?
I would just say simply from the publications that are out there, the local publications, there have been quite a few neurological presentations associated with HIV co-infection. I mean, those ones that Mo had pulled out from the literature, it was mainly the neurological presentation that seemed to be the common ones, and that's certainly in the international literature. It seems to be more commonly associated with the that HIV, the complication more commonly associated with HIV is the neurological one.
Okay, thanks very